So today's topic is effects on auxin on growth response. Okay, so this is our topic for today. Actually, we're going to cover two subtopics. All right. So in the previous lesson, I hope you already watched my recorded video. On the other day, you have this physics webinar, right? So I already discussed about the phytohormones, and there are five major phytohormones, and one of them is auxin. All right. So the roles of auxin in plant response. Let's review back what is the function of the auxin. So auxin, it's functional in phototropism and also geotropism. And this is what we're going to discuss, or what I will explain it today. And auxin also stimulate the cell elongation at the shoot tips. Okay, so having said that, shoot tips, you know, shoot will grow upwards. Shoot, whenever there is an auxin, it will grow upwards. Okay, grow upwards. Okay, that is auxin. Here, how this auxin play important role in grow of a plant. So before that, you need to know where is the calyptil. Okay, calyptil. So this is a seed. Okay, this is a seed and this is a root. And then here is the shoot. So shoot, actually there's a division in it. This is what you call a true leaf, where it will grow into leaves. And then this calyptil part, okay, calyptil, not calyptil, calyptil. This calyptil, you will heard these terms when we discuss about auxin, because auxin will be synthesized in the calyptil. Okay, calyptil is in the shoot, and then auxin will be exist in the shoot because it will be synthesized, because auxin not will be because auxin is being synthesized in the calyptil. So di mana calyptil, you need what my point here is, you need to have the idea where is the calyptil at. Calyptil is at the shoot. Okay. Next. Coba. Macam ada. Okay, someone. I need to admit someone. Okay. So next, we continue. Okay, so calyptil is at the shoot. So this is the picture. Calyptil is over here. And why we need to identify where the calyptil is? Because auxin, the hormone, is being synthesized at the calyptil. Okay, next. Since we already know that calyptil, auxin will be synthesized at the calyptil, there's three experiments to prove that. Facts, whether is it true that calyptil contain auxin? Okay, and then is it true when the auxin is available at the calyptil, is it true this auxin can help cell elongation? So there's 3 experiments to prove that. Okay, so we have this diagram is in your textbook. You can refer it simultaneously sambil dengar teacher explain, kamu boleh tengok. It's on page 104. Okay, so let's start with experiment 1. Let's start with experiment 1. Wait for a minute. Okay. So experiment 1 shows here, this is the shoot and then the tip is cut off. The tip is cut off. So meaning that it wants to show you the calyptil tip, the tip of the calyptil has been removed. Okay, and then after 2 hours, no growth is observed. No growth is observed. And then after 2 hours, they put it back the tip. They put back the tip and then it shows a growth process. The calyptil grows upward. Maksudnya apa yang dia remove tadi tu, the tip itself play important role because the tip has a auxin. Okay, next we go to the second experiment. At the second experiment, the situation is the same where they cut off the tip. They cut off the tip, ni di hujung ni, and then what they do with the tip, they put the tip of the calyptil, they place it on a agar block. Agar block ni maksudnya agar lah, block tu kan bentuk macam ni. Alamak. Okay, bentuk macam ni. Sorry. Okay. Okay. Ini maksudnya block. So this block is made up of agar. Agar-agar lah, agar. Okay. After 2 hours, the agar block is placed on the cut calyptil. The calyptil grows upward. Okay. After 2 hours, they put the agar. Bukannya tip, they put the agar. And then the calyptil grows upward. Maksudnya, okay, teacher terus buat kesimpulan lah di sini. So meaning that whatever in the tip of the calyptil, maksudnya apa-apa yang ada di sini ni, this tip of calyptil contain auxin. And since this is agar, the auxin in the calyptil will diffuse into the agar. Okay, this is the agar. And then this is where the example kita boleh guna ni. Okay, this is agar and this is the tip of the calyptil. They put the tip of the calyptil on the top of the agar. And then auxin in the calyptil, tip of the calyptil, the auxin will diffuse into the agar. So the take of the tip of the calyptil, now the agar itself contain auxin. Okay, the agar block, not the calyptil. The agar block contain auxin. So they put this agar block that contain auxin, they place back the agar block on the cut calyptil. Maksudnya calyptil yang sudah tiada tip lah. Okay, yang tiada tip. Bila dia letak, when they place the agar block, it shows a growth process. The calyptil growth upwards. Kenapa dia boleh growth? Because the auxin from the agar. Auxin from the agar helps the calyptil to grow upwards. Okay, that's the second experiment. And then the third experiment. Tengok betul, third experiment sama juga, they start with the tip is cut off, and then the second one sama juga, they put the tip of the calyptel, they place it on top of the agar block. And then what do we expect to happen? Auxin from the tip, auxin yang teacher tanda merah ni, let's say that's a represent auxin, will diffuse into the agar block. Okay, so this agar block contain auxin. But this time, this agar block is placed off center. Maksudnya dia bukan tengah-tengah macam tadi. In the previous experiment, experiment number 2, they put the agar block at the center. So that's why the calyptil growth upwards. But this in the experiment 3, they put the agar block off the center. Maksudnya dia di tepi sini. So only this part of this calyptil will receive the auxin. Okay, that is why after few hours it shows the calyptil growth bend. It bends. The calyptil growth, calyptil bending. It curves. Sebab because only one side of the calyptil receive or get the auxin. So what it wants to show here, what it wants to show to you here is the effect of auxins. So whenever there's auxin available, the shoot of the tip will grow. Boleh? Okay kah? Okay kah kelas?
Okay.
Sedihnya. Tidak tahu teacher yang sedih ke atau kamu yang sedih. Okay. Please respond sebab bila teacher share my screen, I cannot see you all. So please respond through voice je lah. Teacher tak boleh baca chat. Alright. Now we go to the conclusion of that experiment. Okay, so conclusion, I just mentioned the conclusion. So conclusion is synthesize. Okay, first, the auxin is synthesized at the calyptil tips to stimulate elongation. That's the fact that we want to understand. Okay, and then the second one, okay, this is the important. Auxin distribution influence the direction of the shoot growth. Okay, this has been shown in experiment 3. During the experiment 3, it shows you what happened when the agar, okay, let's say this is the agar, it been placed off center, di tepi dia ni. And then the auxin, meaning that auxin in the agar diffuse into the calyptil shoot only at the, let's say now the agar block, this is agar block, is placed on the left side. Okay, so the auxin will be only diffused or available on the left side of the calyptil. So that is why when the growth happen, only left side will grow. So that is why the calyptil will bend. Okay, calyptil will bend. So side yang ini sahaja yang akan growth available. Okay. So having this idea, now we can understand what is the roles of auxin in phototropism. Okay. Kita tengok next. Okay, before that, effect of high auxin concentration. Auxin, we know auxin is to stimulate cell elongation, right? But if there's a case there is a high concentration of auxin, it will cause two situation. High auxin concentration. Okay. Now we are talking about high, tinggi. High concentration of auxin will stimulate the cell elongation in shoots. Okay, cell elongation in shoots. That is why I use a green color. It shows to stimulate the cell elongation, but most important where it will stimulate cell elongation in the shoots. But high auxin concentration will inhibit, okay, they suppress, inhibit. That is why I use the red color. Show it will stop, inhibit the cell elongation in the roots. Okay. So with this understanding where the effect of high auxin concentration, then later on we're gonna use this idea in geotropism. Nanti kita tengok lah geotropism. Okay, so you need to understand first, high concentration of auxin will stimulate cell elongation in the shoots, but high auxin concentration will inhibit the cell elongation in the roots. They are vice versa, bertentangan. Okay, that is why this is the reason why we cannot see shoot will. Kenapa tiada shoot yang grow upwards? That's the reason. Kita tengok shoot sahaja yang grow upward, tapi kalau, okay, let's say this is the plant. Okay, this is shoot. Kenapa shoot dia boleh grow upward? Because of this fact. High auxin concentration will cause cell elongation in shoot. Tapi high concentration in the root will inhibit the cell elongation of the root. Sebab itu kenapa kita tidak nampak shoots in plants tu naik macam tu, tidak. It will goes into the soil. Alright. Any questions?
So far, no.
Okay, thank you. The next, the roles of auxin in plant response. Okay, apa beza dengan yang tadi? Just now, we want to know the auxin, roles of auxin, function of auxin in growth. Whether the plant grow or not. Now is the response. We already learned about response in plant. Okay, as maybe you can recall it as tropism and nesty. Okay, kita plant response. And then for auxin, the plant response, auxin is play important role in phototropism. Okay, what is phototropism? What is phototropism, class?
Plant respond to light.
Yes. How the plants respond towards light. Okay, thank you. I think that is C4. Betul kah? C4 kah tu?
Yes.
Okay. So thank you, C4. So the phototropism is how a plant responds towards light. Okay. So having said that, I try to give you a situation here. So let's say this is a light. Okay, this is a light. Saya mau juga lukis light ni. Okay, this is a light. Sunlight lah, of course. And then this is your plant. This is your plant. So, whenever the plant is exposed to light from all direction. Okay, sekarang ni dia expose kan from all direction, meaning the whole plant is exposed with light towards exposed with light. Okay. So they expose so that the auxin will uniformly distribute. Okay, apa maksud ayat ni? Tadi kita tahu auxin dia akan ada di at the tip of the shoot. Okay? And then when exposed with all direction of the light, jadi auxin dia akan distribute uniform. Uniform maksudnya seragam, meaning the whole region of this plant will receive auxin. Semua tempatnya akan ada terima auxin, and then when there's auxin available, it will grows. Okay, it will grows upright. Okay, and the second situation is when uneven expose of light. Maksudnya, okay, when this is a source of light and then this is plants. So the plants is exposed only at one side. Ini yang penting dengar betul-betul. Okay, ini yang biasa kita faham dengan memang logik lah. Kita akan nampak the plants growth upwards because the auxin will distribute uniformly ataupun will uniformly distribute. But if this case happen when the light, the source of light ataupun the exposure of light is only at one side. Let's say here is left right. This is the right side and this is the left side. Okay, so this right side it receive a sunlight exposed to the light. This left side we call it as shady atau shaded, yang gelap. Left side is shaded, right side is a bright side lah, yang bagian cerah. Okay, why it is important to identify which is bright, which is shady side? Because auxin is photophobic. Okay, auxin, they are photophobic. Apa maksud photophobic?
Tidak suka sunlight.
Yes, it doesn't like a sunlight. So, bila tidak suka sunlight, this auxin will move away from the sunlight. It will go to the shady area, shaded area. So di sini lah the auxin will accumulate here. Okay, so dia akan sini lah. Nampak dia akan berkumpul di sini. It will accumulate to it will move to the shady area. And then what happened? Kita sudah belajar tadi sebelum ni, bila only one side, meaning that only the left side of the calyptil, siapa yang ada auxin, dialah yang akan grow. So this left side will grow. So bila grow, so it looks like it will grow towards light. Boleh?
Boleh.
Okay, so okay, thank you for responding. So here the key point here I want to tell you is auxin is a photophobic. So it will go to the shady area. So shady area has a benefit. They got auxin, so the shady area will grow. So there's an uneven growth lah. Maksudnya yang tiada auxin tu tidak akan growth. It won't elongate, but the shady area will elongate. That is why it shows a curve growth. Okay. Now we go to this diagram is in chapter it's on page 105. So this more like experiment lah to prove that fact, to prove that auxin role, how auxin plays an important role in phototropism. Okay, so di atas ni adalah before, di bawah ni adalah after. Okay, so let's look at before situation dia lah, before 2 days before the reaction happen. Okay, situation here you can see light is on the left side. Okay, the sunlight is over here, meaning that the source of light is only on one direction. Dia bukannya betul-betul di atas. It's on one direction. Okay. So when the light is only at one direction, we're going to expect this collaptil growth bending. There will bending at the collaptil. Dia akan ada bengkok. Okay, so ini adalah beberapa anak pokok, beberapa seeds. Okay, this name it as inilah A, B, C, D, E, F, G. Okay, 2, 3, 4, 5, 6, 7 seeds. Okay, and then they manipulate the situation. For the seed A, control, meaning they didn't do anything. They didn't do anything with the seeds. Seed B, they cut the collaptil, the tip of the collaptil. They cut the tip of the collaptil. C, they covered the tip of the collaptil with opaque cover. What is opaque cover? Opaque ni means it not transparent, where the light cannot get through. Okay, cahaya tidak boleh tembus. And D, the collaptil is covered, but this time with a transparent cover. So having said that, transparent cover, we know it will let the light to pass through. Okay, akan ada cahaya boleh lalu di sini. Dia boleh terima cahaya lah on the seed A, D. So for E, this time the base of collaptil tip is covered with opaque object. Sama juga opaque object, non-transparent, tapi at this time they put it on the base, di bawah, bukannya betul-betul di hujung. Okay, F, the collaptil tip is separated by a piece of agar block. It's been separated, meaning maksudnya dia cut lepas tu dia tutup macam tu lah. Okay, dia letak tengah-tengah, in between there is a agar block. And G, the collaptil tip is separated by mica. Okay, mica ni dia adalah satu elemen stone, macam batu lah. Okay, so okay, so in this agar block, we expect that we know the tip of the collaptil contain auxin. So maksudnya di sini ini ada auxin dia. Yang B tidak auxin, sudah kena buang. Yang C, mana C pula? Yang C collaptil dia masih ada, cuma kena tutup. Yang D pun masih ada auxin. E pun masih ada auxin. Yang ini auxin dia masih ada, yang ini pun auxin masih ada. Okay, tengok betul-betul lah. After 2 or 3 days, we look at the results. Okay, yang A, it will bend towards the light. It will bend towards the light. Sebab apa? Ini kan facing this side facing the light. So meaning that shady area dia sekarang sebelah sini. So mana yang shady area, the auxin is photophobic, so it will move to the shady area, so that shady area will grow bending towards the light. Okay, ni basic idea yang kamu kena ada. So B, that is why B, walaupun dia ada shady area di sini, tapi there is no auxin because the collaptil has been removed, has been cut off. Sebab itu, no growth is observed. Itu reason dia. Tiada di sini. No, auxin not available. Okay, C. However, C, decalyptyl, dia tidak bending. Decalyptyl grows upward. Why is that? Remember, auxin is photophobic. Bila photophobic, dia suka gelap. Dia fobia cahaya. So, dia suka gelap. Bila tutup pun, the tip of the calyptyl is covered with opaque cover. So, it creates a shady region, the whole tip. So, okay, the whole tip is become a shady area, shady region. So, dia tidak akan move ke mana-mana. Okay, so this auxin will grow upward. Okay, D. So, for D, walaupun tutup, it doesn't create a shady area for the whole calyptyl. So, shady area dia sama macam yang the first one, dia berada di sini. So, that is why this calyptyl bend towards the light. Okay, it grows towards the light, bending. E. Kalau E, okay. E kita cakap ada auxin. So, auxin, as long ada auxin, dia akan bending. Itu je kamu kena ada idea. Tapi ini kenapa dia letak manipulator, dia letak di sini, there's an opaque object. But it was put at the base, bukannya on the top, at the tip. So it shows that the auxin is available at the tip of the calyptyl, not at the base. Okay. Next, F. Okay. Even though there's an agar there, tapi kita tahu tadi agar ni, the auxin can diffuse. Okay, it will diffuse, this auxin, it will diffuse through the agar block. Then yang sebelah bahagian bawah ni, it will receive auxin. That is why the cell can elongate. Bila elongate, dia akan towards the bending. Kenapa dia bend? Sebab dia ada shady area sini. Okay, shady area ni. Okay, however, on G, even though dia ada auxin and dia ada shady area, tapi no growth is observed. Why is that? Because this mica block the auxin. Auxin yang berada atas at the tip of the calyptyl cannot pass through. So the cell cannot elongate. Sebab itu no growth is observed. Okay, boleh?
Boleh.
Okay. Thank you. I think that's Aisha voice. Okay, and Sefo. Okay, so conclusion dia apa yang kita mau tunjuk, again and again it wants to shows you that the auxin is responsible for phototropism. Because, why phototropism? Because this auxin is photophobic. Memang lucu lah juga kan, dia photophobic, tapi dia lah yang menyebabkan plants to bend towards the light. Okay, tapi that's the point. Dia kena jadi photophobic, barulah ada movement, baru ada respon. Okay? That's the conclusion for this experiment. Mesti kamu kena ingat, auxin, photophobic, move to shady area, and then shady area will grows and atau cell elongation happen at the shady area. Okay, next, the roles of auxin in geotropism. Okay. Tadi phototropism, sekarang geotropism. Bila cakap geotropism, what is geotropism? In anyone?
Respond toward gravity.
Yes, it's a response towards gravity. Okay, so geo means earth, so it's towards response towards gravity. So auxin, again, auxin memang kerja dia adalah untuk cell elongation saja lah. Adalah kerja dia lain, tapi in this maksudnya ada function yang lain. Ini kita sudah bincang dalam phytohormones. Tapi in this topic, you need to understand that auxin is stimulate the cell elongation. Okay. And just now I told you high concentration of auxin. Okay. High concentration of auxin dia ada dua. Dia ada dua. The first one, it will stimulate, and at the same time it will inhibit. Stimulate who? Stimulate the cell elongation of the shoot. Tapi kalau tinggi auxin, it will inhibit the cell elongation of the root. Okay. So maksudnya ini. Okay, nanti kita tunjuk lebih detail apa maksud ayat ni. And then what else? Okay. So with this idea, high concentration will inhibit will inhibit the cell elongation in roots. Because when we say about geotropism, automatically we will think about roots. Right? Sebab roots yang lebih dekat dengan gravity. So before we discuss about before we discuss about auxin in geotropism, you need to recall that geotropism, there are two types, which the first one is positive and the second one is negative. When we say positive geotropism, meaning that the plant growth towards ataupun follow the gravity force. Tapi kalau cakap negative geotropism, the plant shows a response where they're moving away from the gravity force. So usually positive geotropism shown by okay, bahagian mana plants yang shows the positive geotropism? Shoot ke root? Bahagian mana yang, which part of the plant shows a positive geotropism? Is it shoot or root?
Root.
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 Okay. Oh boy. Sudahlah teacher kasih high volume my earphone ni. Okay. So good. Is the right answer. Geotropism, positive geotropism shown by root. So opposite on that, negative geotropism shown by shoot. Sebab kita tahu shoot dia akan upwards, away from the gravity. Gravity kan on the underground. So kalau root dia akan tumbuh di bawah. Okay. So kita sudah ada dua idea tu. Satu about the high concentration of auxin, it will inhibit the cell elongation in the root. And then the second important point that geotropism, there's a positive and negative one. Okay. Let's look at this one. Okay, tengok dulu. This is, apa ni? Apa benda ni macam Super Mario punya cendawan ni? Seed, yes.
Seed.
It is a seed. Siapa warna merah? It will be a proper one. Okay. This is a seed. The color, this is seed of bean, I guess. Kita sebagai contoh lah. Okay, this is a seed and it will goes horizontally. Okay, it will goes horizontally. It will grow horizontal. And then there is a grow of shoot and root. Okay, shoot and root. Ini betul-betul yang starting. And then we know on the ground there is a gravity atau gravity force ataupun pull of gravity. It's either way, which term do you want to use? Pull, uh, gravity pulls ataupun pull of gravity ataupun gravity force. Mana-mana lah. So, itu logic. Seed, kita tahu di dalam tanah, it's in the soil. And then there's a existence of gravity pulls. Okay, why is important is that because of this gravity force ataupun gravity pulse, it will pull the auxin. Pull dulu lah. Pull dulu. Dan kenapa? Sebab nanti ada perkataan pulse. In your textbook, they use the term pulse. Kolam. Pulse. Pulse pula. Pulse. Sabar je lah. Bukan pulse, pulse. Pull dengan pulse yang ini, kolam. Now I'm talking about pull, tarik. This gravity will pull the auxin. Kan ada auxin di sini. Okay, auxin di hujung, at the tip. Tapi because of the gravity pulls, this auxin will accumulate at the bottom side. Okay, when I say bottom side, okay, tengok di sini ya. Ini adalah, kita zoom in, ini shoot. Kan dia ada, ini kita panggil dia upper. Ini lower ataupun bottom. Okay, guna istilah buku ni lah, bottom. So, bottom ni maksudnya yang di bawah. Kalau if you can observe closely in your textbook, di bahagian bawah tu yang ada dot hijau tu. That's what it means by the high auxin concentration at the bottom. Okay, kenapa dia ada di bawah? Because of the gravity pulls. Okay, so this gravity cause the auxin to accumulate at the bottom of the shoot tip. The same things happen at the root. Because the gravity pulls the auxin so that the auxin accumulate at the bottom side of the root tip. Di bahagian bawah sini. Okay, this is the bottom, this is the top. Top, bottom, upper, lower, kan? Okay, so bottom of the root. Dua-dua benda yang sama berlaku. The same thing happens at this both side. Root dengan shoot ni dua-dua dia punya auxin has been pulled at the bottom. But kita sudah belajar tadi, high concentration of auxin cause the cause the cell elongation at the root, at the shoot. Tapi high concentration of auxin will cause the inhibit the cell elongation at the root. Okay, that is why the shoot will bend upwards. Bahagian bawah dia ni akan grow upwards. Tapi yang ini dia inhibit. So sebab itu kenapa dia tidak jadi macam ni. Mana teacher mau lukis ni? Okay, sini. Dia tidak jadi macam ni. Ini seeds, ini shoot, ini root. Kenapa dua-dua tidak naik? Sebab itu kenapa tidak berlaku macam tu. Kenapa satu pergi atas, satu pergi bawah? Because of this auxin. Boleh?
Boleh.
Okay. Now, ada mau respon lagi? Okay. Now that tadi kita bilang high auxin concentration kan. Lepas tu barulah kita pergi. So this is what we call geotropism. The shoot shows a negative geotropism sebab dia tidak ikut gravity, it against the pull of the gravity. Tapi this root, it grows downward, so it follow the pulse of the gravity. So it shows a positive geotropism. Boleh? Tu je soalan, boleh? Okay. Okay. So this is a video to shows you, this is time lapse. Time lapse tu maksudnya time lapse dia ambil ada certain time yang kita tengok dia ada movement. Okay, this is a seed and it wants to show you the growth of shoot and roots. Okay, watch carefully. Okay, that is a root coming bukan coming off, it's come out from the seed. It already grows, show a growth, and it grow downwards. And then there's a shoot coming out, and then it will grow upward. Why is that? Because the auxin. Nanti dah ada leaf and ada now the formation of roots cell. Ni teacher akan share dalam apa nama dia ni? Teacher akan dia kena siram baru dia basah tadi kan, dia kena siram baru dia sambung tubuh. Okay, that is the time lapse of seed germination.